After studying this module, you shall be able to know about the significance of centrifuges, the basic principle and methodology of centrifugation, the types and application of centrifuges. Centrifugal force is a combination of two Latin words, centrum meaning center and fugere means to flee. It is the ostensible force that draws a gyratory body away from the center of rotation and is generated by the inertia of the body as the body's path is constantly redirected. The centrifuge is a significant instrument in cellular and molecular biology research laboratory. Basically, all centrifuge instruments consist of a motor which spins a rotor holding the experimental sample. It is predominantly used to segregate particles or macromolecules like cells, subcellular components like nucleic acids and proteins on the basis of their sedimentation rate. The sample gets separated due to variation in their sedimentation rate because of their different shape, size, viscosity of the medium, density and the rotational speed of the rotor to separate. In 1925, Theodore Swedenborg designed the first analytical ultracentrifuge. The vacuum ultracentrifuge was developed by Edward Graydon Pickles. Vacuum systems similarly facilitated the upkeep of constant temperature. In 1946, Pickles co-founded Spinco Specialized Instruments Corporation and marketed an ultracentrifuge based on his design. In 1949, Spinco presented first preparative ultracentrifuge to attain a maximum speed of 40,000 rpm. In an analytical ultracentrifuge, a sample being whirled can be examined in real time through an optical detection system by means of ultraviolet light absorption and interference optical refractive index sensitive system. The types of information that can be achieved from an analytical ultracentrifuge consist of the gross shape of macromolecules, the conformational change in macromolecules and size allocations of macromolecular testers. Principles of centrifugation. The centrifuge encompasses principle of sedimentation where the acceleration at centripetal force reasons denser constituents to isolate out besides the radiating direction at the bottom most of the tube. With a similar theory, lighter substances will be likely to move to the top of the tube. Particle with more density than the solvent residue and the particle with lesser density, then it drifted the solution to the top. The greater the difference in density, the faster they move. In Isopycnic conditions, that is, where there is no difference in density, the particles remain stable. To take benefit of even minute differences in density to separate different particles in a solution, gravity can be substituted with much more potent centrifugal force delivered by a separator. According to the sedimentation theory, sedimentation is the affinity of particles in suspension to settle down in the fluid where they are pulled and come to rest against a barrier. This is on account of their motion through the fluid in response to the forces acting on them. These forces are due to of gravity, centrifugal speeding or electromagnetism. The value for a molecular sedimentation velocity in a centrifugal field is called as its Swedenborg constant or S. You can see the calculation of S expression given here, where m is the molecular weight, S is the Swedenborg coefficient and V bar is the partial specific volume of the molecule, N is the Avogadro's number and F is the frictional coefficient. Methodology. Centrifuge makes use of density difference among the particles or macromolecules and the medium where they are spread. An artificially induced gravitational field is exposed for the dispersed systems. A centrifuge is generally propelled by an electric motor which sets an object in rotation around a stationary axis, applying a force vertical to the axis. Procedures for centrifugation characteristically specify the amount of acceleration to be applied to the sample, relatively then postulating a rotational speed such as revolutions per minute. This peculiarity is significant since two rotors with diverse diameters running at the similar rotational acceleration will subject samples to different accelerations. Throughout circular motion, the acceleration is a product of the radius and the square of the angular velocity, and it is conventionally known as relative centrifugal force. The acceleration is measured in multiples of g, which is the standard acceleration due to gravity at the Earth's surface. Let us assume an element of mass m in a centrifuge tube filled with a liquid. The particle is acted on by three forces: Fc, centrifugal force or relative centrifugal force; Fb, buoyant force; Ff, frictional force between the particle and the liquid. The particle in a centrifugal field will experience a centrifugal force defined by the product of mass of the particle, distance from the axis, and the square of angular velocity. Types of centrifugation. Differential centrifugation. This technique is occasionally speak of differential sedimentation and is fundamentally a method of successive centrifugation with increasing centrifugal force. Separation is mainly dependent on particles' mass and size, where heavier particles or cells settle first at lower g values. However, in various cases, differential centrifugation is used to separate out intracellular matter, and consequently, this method is important for purported subcellular fractionation. But to obtain a pure organelle fraction from differential centrifugation is practically impossible. Moving boundary or zone centrifugation. In this, the whole tube is occupied with sample and centrifuge. With centrifugation, parting of two units can be achieved, but any unit in the combination may result in the supernatant or the pellet, or it may be distributed in both fractions. However, it depends upon the size, shape, density, and condition of centrifugation. The mixture of all of the sedimented components is known as pellet, and it is mixed with unsedimented particles of any kind in the bottom of the tube formerly. The only constituent which is refined is the slowest sedimenting one, but its yield is habitually very little. The two portions are recuperated by transferring the supernatant solution from the pellet. The supernatant can be recentrifuged at greater speed to obtain extra purification with the formation of new pellet and supernatant. Rate zonal centrifugation. In this, we keep the sample on the topmost position of the tube in a very thin zone of density gradient in the influence of centrifugal force. The particles will start sedimenting over the gradient in distinct zones as per their size, shape, and density. The run must be terminated before any of the separated particles reach the bottom of the tube. Particles of the same size but different shapes will distinct. The particles having more frictional coefficient will transfer slower. This technique is called velocity gradient centrifugation. Isopycnic or density gradient centrifugation. In isopycnic technique, the density gradient column encompasses the whole range of thickness of the sample material. The matter is consistently agitated with the gradient material. Each particle would residue at the point where the gradient density becomes equal to its specific density in the centrifuge tube and remains constant. Isopycnic technique separates particles into separate zones exclusively on the source of their density differences. For example, the gradient may possibly be of such density range that one component sediments to its density in the tube and remains there, while other component sediments to the bottom of the tube. The self-generating gradient technique over and over again entails long hours of centrifugation. Types of centrifuges. Broadly, centrifuges are classified into two categories: laboratory centrifugation and preparative centrifugation. Laboratory centrifugation. They are used for small-scale separation and clarification. Characteristic liquid capacities controlled by such devices are in the range of 1 to 5000 ml. The solid to be centrifuged is dispersed into suitable numbers of centrifuge tubes that appears as test tubes, which is joined in a symmetric method to a revolving block known as rotor. There are two kinds of rotors: fixed angle rotors and swing out rotors. The fixed angle rotors grip the centrifuge in an immovable way at specific angle to the axis of rotation, whereas swing out rotors clamp the tube analogous to the axis of rotation. Though the rotor is immobile, but when the rotor comes in motion, the tubes blow out in a way that they are aligned perpendicular to the axis of rotation. Preparative centrifugation. Preparative centrifuge can handle considerably more liquid capacities as compared to the laboratory centrifuges, which characteristically fluctuating from unit liter to some thousand liters. Its various types are discussed further. Tubular bowl centrifugation. The suspension to be separated is put into these devices from one end and the supernatant and precipitate are taken out from the other end of the device in a continuous or semi-continuous manner. At any point, the motion of a particle within a tubular centrifuge in the radial direction is governed by the following equation. The force exerted on a particle is equal to the frictional force suffered by the particle. The particle will settle down because to centrifugal acceleration till the two forces are balanced. Disc stack centrifugation. The disc stack centrifuge is a different type of preparative centrifuge that is compressed in design and provides better solid liquid parting in comparison to the standard tubular bowl centrifuge. The particles are thrown outwards and these come into contact with the angled disc stack. Once this takes place, the disc is slid down and the product is collected at the edge of the bowl and discharged from the device in the form of slurry. The liquid flows up the device along the central region and is discharged from the top. Chamber bowl centrifugation. The chamber bowl with intermittent discharge has no disc stack. The chamber bowl is like a tubular centrifuge but with insignificant L by D ratio. It is appropriate for moving viscous feed or more concentrated solid feeds. As discussed, the clarification capacity is less than that with the disc stack centrifuge due to a reduced settling area. Though in comparison to disc stack centrifuge, the chamber bowl can take higher feed solids. The feed rate usually is smaller for chamber bowls compared to that of disc stack centrifuge for the same ball size. Ultracentrifuge. An ultracentrifuge is an extraordinary type of centrifuge where the rotor revolves at a much higher speed than a standard centrifuge. Typically, the rotation speed in ultracentrifuge ranges from 30,000 rpm to 50,000 rpm. An ultracentrifuge is usually used for separating macromolecules from solvent or for fractionating mixtures of macromolecules. Ultracentrifuges are developed for analytical as well as for preparative applications. An analytical centrifuge is largely used for examining the properties of macromolecules besides for analyzing the complex mixture of macromolecules. Preparative ultracentrifuges are employed to purify macromolecules such as proteins and nucleic acids based on their physical properties such as size, molecular weight, density and mobility. The high rotating speeds used in ultracentrifuges can generate substantial amount of heat requiring cooling measures to be attached with these devices. Now we will see the application of centrifuges. Separation. Centrifugation is employed for solid-liquid isolation in a condition that the solids are denser as compared to liquid. It can be applied to isolate a heavy phase and two lighter liquid phase, in which one will be lighter phase being lighter with respect to each other. As stated, solids can be lighter than liquid and separation is by floating of the dispersed solid phase. Interpretation. Centrifuge is used to segregate the discharge separated lighter liquid phase. The purpose is to reduce the discrete suspended solids in the light continuous phase. Usually, only fine submicron biosolids are left uncaptured by centrifugation and they leak with the discharged light phase. Degritting. Degritting is like classification where surplus particles larger or denser are rejected in the sediment with smaller or less dense products overflowing in the light liquid phase. Another condition is where smaller surplus particles are excluded in the light liquid phase and valuable higher solids are settled with the higher phase. Thickening or concentration. In order to concentrate the solid phase by sedimentation and compaction, removing the excess liquid phase in the overflow or centrifuge devices are regularly used. This decreases the volume of the product in subsequential processing. Separation and re-pulping. With the concentrated suspension containing impurities such as salts and ions, it is diluted and washed so that the impurities are dissolved in the washed liquid. Afterwards, the suspension is directed for centrifugation to remove the expended washed liquid with dissolved contaminants or finely suspended solids. Subsequently, the product can be further concentrated by centrifugation. Centrifugal force, word from Latin centrum meaning center and fuga means to flee, is the ostensible force that draws a gyratory body away from the center of rotation. It is produced by inertia of body as the body's path is constantly redirected. The centrifuge is an important instrument in cellular and molecular biology research. The centrifuge encompasses principle of sedimentation where the acceleration at centripetal force reasons denser constituent to isolate out besides the radiating direction at the bottommost of the tube. With the similar theory, lighter substances will be likely to move to the top of the tube. According to sedimentation theory, sedimentation is the affinity of a particle in suspension to settle down in the fluid where they are pulled over and come to rest against a barrier. This is due to their motion through the fluid in response to the forces acting on them. These forces are due to gravity, centrifugal speeding or electromagnetism. Centrifugal evaporators are designed to evaporate multiple samples down using a combination of centrifuge, rotor, vacuum pump and solvent trap. The centrifuge chambers can have the option of heating also. A refrigerated laboratory centrifuge applies an inbuilt cooling system in order to retain the centrifuge chamber, and thus the samples being separated is already set under ambient temperature. Centrifugation in an unrefrigerated centrifuge generally makes sample to warm up to a certain degree and temperature delicate samples could be affected.